Okay, now though, for the other timer, it's going to be, you have 15 minutes total. Most talks have 15 minutes. 8 minutes is going to turn yellow. That means you have 2 minutes left. And then when it turns red, it means questions are starting. Okay, and then 5 minutes for questions. You got that? And I think that's all my, I think that's everything I was supposed to say. Now, Sue. Also, welcome to Stowers, and welcome to Biocuration 2025.
Okay, so I am giving the official ISB welcome for everybody. So welcome. Thank you all for coming. This has been a long time in the planning, so it's really great to be here. It's great to see you all. Let's see then. All right. Yes.
This year.
Oh, yeah, that.
I have a clicker. Okay, there's a clicker.
Oh, yeah.
Thank you very much. Okay, so we just wanted to say a little bit about the ISB. So as of just a couple of years ago, we are now officially a Swiss nonprofit organization for biocurators, developers, and researchers with an interest in biocuration. We try to promote the field of biocuration, the work of biocurators, and then also I think a big part of what we do is making connections between biocurators, making the free exchange of information, really helping people build a network and communicate so that when you have a problem, you have a community to lean on. And then we've supported some local meetings, and every year we support our international meeting. This year it's in the US, and it moves each year, and we'll have the announcement at the end of where the next one is. Yes, and then we support different awards. But so this year we are in the US, but we've rotated. So this is our 18th annual meeting. We move between the Americas, Europe and Africa, and Asia and Oceania. So in 2023, we were in Padua, Italy. In 2024, we were in Hyderabad, India. So we try to move, and then ISB during the meetings. We would love to talk to people about joining ISB, about what we do, about how we support our community, and how you can get involved. We'd love to get more involvement, whether it's just involved in community, in committees, or whether you actually want to be on the EC. We communicate through Blue Sky, and that's our Blue Sky handle. We communicate through Mastodon, and we really communicate through Slack. Somehow that has become a big channel for communication. So if you didn't get the ISB Slack join from the last one, we got it up here now, and feel free to follow us on either Blue Sky or Mastodon or both. There are actually a number of EC members at the meeting. I don't know if I could get the ones who are in here now, maybe to stand up and just wave and say hello. So I am the current chair. We have Charlie, Tarcisio, Sonia, and Victoria are in the room. I thought Mark was supposed to be here, but he may not have made it. Hopefully he will get here. ISB gives a support presentation. So every 2 years, we have an award that's given for exceptional contributions to biocuration, the Lifetime Achievement Award. The last running of that was in 2023, and we had a tie. So as part of this, we always have them give a keynote at the Biocuration conference. And we decided that we had a tie. We weren't going to do a tiebreaker. We would just, Pascal presented in the India conference, and Sandra is presenting here. We also give out a number of travel grants. And then our update on the 2026 meeting will come at the end of the session. But so just to let you know, when you do join the ISB and you give us dues, this is some of what your dues go to. It's helping people who otherwise couldn't afford to get to the meetings to come, and it's giving awards to recognize the work of your colleagues. And this year we decided to create a new officer position on the EC. which is our EDIA chair, so for equity, diversity, inclusion, and accessibility. And I have to say, we're really happy. This is the first time we've managed to have a fully hybrid conference, and it seems like it's going pretty well. And I want to recognize Luana, who is volunteered to be our first EDIA officer. And so she's the point person. If you have concerns, if you have questions, she's the point person to ask. And then I just want to close, keep this short, say I hope everybody has a great meeting. Come say hi to us, talk, if you have questions about what ISB does. If you want to know more, we're all, I think, fairly friendly and easy to talk to. So thank you all.
Hi, good morning, everyone, and welcome to the Stars Institute for Medical Research. My name is Alejandro Sanchez Alvarado, and I am an investigator here at the Institute, running a lab, but I'm also the Institute's president and chief scientific officer. And Sophia asked me to give a couple of remarks before the beginning of this very exciting meeting that you are all here gathered to do. And so what I thought I would do is to tell you a little bit about our mission, and then tell a little bit about why we think that this is a very important event for the future of biomedical research, all right? So the first thing I want to share with you is the picture of our founders. These are Jim and Virginia Stowers standing at this site about 25 years ago when they were digging a hole to build this building. This was a dream they've had. They're both cancer survivors, and they were also founders of American Century Investments, which is a large mutual fund company. It used to be, and now it's an investment company that is right here in Kansas City, Missouri. Is a private institution. Now, the reason why I bring you all this to your attention is because they came up with a truly innovative dream that they have been now executing for these 25 years, which is that as majority owners of a non-publicly traded investment firm, they decided to donate all of their earnings, all of their ownership of that company to the Institute. So the Institute is actually majority owner and controls the board of a for-profit company, American Century Investments, and we receive about 50% of their profits just to fund the research that you see here. So this is a truly extraordinary, situation. The CEO of American Century and myself like to refer to this as a universe of one, because I don't know of any other institution that relies entirely on its endowment to carry out its scientific mission. And what is our scientific mission? It's probably the best gift that Jim and Virginia gave us is written here. And I'll read it to you because I think it's a fairly unique mission for this institute, which is to make a significant contribution to humanity through medical research by expanding our understanding of the secrets of life, and by improving life's quality through innovative approaches to the causes, treatment, and prevention of diseases. So for the last 25 years, we've been very busy setting up the Institute. And for the last 50 years or so, we have been delving, deliberately and as robustly as possible into trying to reveal what those secrets of life are. And so the Institute characterizes itself by working on a diverse range of organisms, because we believe that nature's probably the best laboratory out there. It's been busy for about 4 billion years. Generating life, and the answers to many of the problems that affect us today as humans have probably been solved multiple times in multiple species across their evolution on the planet. And so if we can reveal those secrets, it should inform what we do here. So what I would like to share with you is the following notion. It's something that I'm beginning to realize is not very commonly discussed. But I'm going to start with these two fellows. You know them. These are Charles and Alfred, Chuck and Alfie, I guess, who gave us the theory of evolution, right? And so this is natural intelligence, gave us natural selection. It was a multimodal approach to data. They collected data in the field, they measured, they combined approaches like chemistry, physiology, biology, and generated, without knowing it, this latent space where they gather all this using their brains and to produce a synthesis that ended up being natural selection. Now we're in the age, of course, of artificial intelligence, and it's It's growing, it's there, can do some terrific things, and it's not very good at making these drawings. I just use DALL-E I ask it, draw me a artificial intelligence, diagram for biology. It's all misspelled. There's all kinds of problems with this image, okay? But it is here. It is with us. It's another tool. We need to learn how to use it. The reason why I bring this up is because I believe that for the most part, at least for almost all the applications that I've seen so far for AI is actually not dealing with natural selection. It's actually dealing with artificial selection. Almost all the tools that I've seen for artificial intelligence apply are for tissue culture, domesticated animals, et cetera, et cetera. And we're hoping that by extracting this biology, that's going to tell us something about biology. I think it's okay, it should be done, but it's not necessarily what I see the most exciting forward aspects for artificial intelligence. So why do I say this? Because domestication really gets rid of a lot of things from organisms. These are the 7 major organisms that we use in the life sciences to study what we study. And if you now look at where these animals reside in the tree of life, and this is relatively old, there's already more phyla in there in the slide. You see that the 5 major vertebrates, are in just one phyla up there at the top of the tree. And then the 2 invertebrates that we have worked on for a long time, Drosophila and C. elegans, they really belong to essentially 2 groups on that 3rd class at the bottom, the so-called Ecdysozoa, right? Now, if you consider there's anywhere between 8 to 9 million species on the tree of life, these 5 or 7 organisms represent approximately, out of these 33 different phyla and counting, 9 times 10 to the minus 5% of all known animal species. So we're clearly undersampling. That's how we see it. We're clearly undersampling biological processes. So why I think this is a problem? The reason why I think this is a problem is because, we have now received a lot of emphasis from funding agencies and prize-winning societies and so forth, that the life sciences really do strive and reward the building of beautiful, logically impeccable, and comprehensively simplified systems. We want to reduce complexity to something that we can really understand. And the work that goes into doing that is rewarded. You get Nobel Prizes for this, you get awards for this, you get grants for this. But this, more often than not, leads astray because they're usually incomplete. And so when you think about how evolution has taken hold on the life forms of this planet, you realize that it's not following any kind of logic. It's really a syncopated rhythm of complex and contingent histories, all of which are shaped by the vagaries and vicissitudes of time, place, and environment. And we rarely take into consideration, as a multimodal approach, except for Darwin and Wallace, the vicissitudes of place and environment. We just assume that all life forms behave pretty much the same way. So in my opinion, I think that these simple laws with predictable outcomes, they cannot fully describe the procession and fortuitous nature of life. And so I'm going to give you an example, and I'll leave it at that. And I'm going to use vertebrates as an example to just tell you what I mean by this. So we know that mammals have common ancestry. They came from one original organism millions of years ago, and they share a similar toolkit, the kinds of things that all of us curate here: genes, proteins, regulatory networks, and the like, right? Now, mammals alone differ close to 1 million-fold in size. That's for the same toolkit. That's a genotypic, phenotypic space that they occupy. Here's the smallest of these animals. It's about 3.5 centimeters long, and that's generous because I think it includes the tail, okay? Just generous. But then if you look at the biggest mammal, which is the humpback whale, you see it right here. It's extraordinarily large, same genome, pretty much, same ancestral genome, very, very different animals, right? Lifespan also differs about 100-fold maximum between mammals, okay? That's what needs to be captured when we try to understand mammals, i.e. us and everybody else that, is a mammal, right? The remarkable thing is that you'll say, well, that's because, the genomes are different and all this other stuff, blah, blah, blah. Well, their genome sizes are almost the same. A number of letters of sequences can codify this vast diversity of biological phenotypic space. That is really astonishing. If we learn anything about sequencing genomes, it's probably this, right? So what I would like to say to conclude is that I think that it would be appropriate for the next decade, I think for the balance of the decade and going forward, to actually take these tools that artificial intelligence is giving us to help us curate these multimodal approaches, images, data, kinetics, etc., and actually start applying it. I think it'd be fire if we could apply it to natural selection. So I'm going to leave you with this. I want to welcome you again to the Stars Institute for Medical Research. Please feel free to roam around, ask us questions, and we'll be more than happy to tell you what we're doing here, which is mostly, as I told you, trying to reveal the secret of life. So with that, thank you very much for being here, and have a fantastic conference going forward. Thank you.
Well, Alejandro gives us a perfect introduction to our next speaker in talking about sampling bias. So our next speaker comes from the Imageomics Institute at the Ohio State University. Thanks. And the whole idea behind the Imageomics Institute is that images represent the most abundant source of documentation of life on Earth. And not just images that are taken in laboratories, but images that are taken anywhere by any device. So what a great way to be able to look and extract features and traits from those images and develop tools to do that. And of course, it brings to mind a whole host of biocuration challenges. So Sam Stevens is here from that institute, and he's going to talk to us on that subject today. At the end, when you have questions, approach the mics. And if you can't approach the mic, raise your hand and a mic runner will come and give you the questions. Thank you.
Got it. Okay. Hi, everyone. Really excited to be here this morning. My name is Sam Stevens. I'm a senior PhD student at the Ohio State University in the Department of Computer Science and Engineering, where I'm advised by Professor Tanya Bergerwolf, who unfortunately couldn't be here today, and Professor Yusu, also in the Department of Computer Science and Engineering. So, yeah, Alejandro's talk was great. It's basically has motivated the rest of my talk, so this is going to be really easy. You know, this title captures our main idea, that images are not just these pretty pictures, but they're this like massive underused data source for understanding the secrets of life on our planet. So I want to talk about how we can convert billions of pictures into actionable structured knowledge. Okay, so the problem obviously is having too much data in some sense, right? We live in this age of data overload. In the biological sciences, data comes in all these different modalities and formats, right? We have genomic data, proteomic data, text-based descriptions, knowledge graphs, ontologies, sensory outputs, and images, which is the one I'm most excited about. And I think one way to think about how these different formats exist is this trade-off between rare structured data and unstructured but abundant data. So hopefully everyone agrees that, you know, often having more data is better than having less data. And often structured data is more useful than unstructured data. But like I said, the problem is you typically have this trade-off. And so I think sort of the two thoughts around, you know, getting abundant structured data is making it easier to generate structured data. Maybe this is making like gene sequencers cheaper or lowering the barrier of adding additional data to your ontologies and knowledge graphs. But the other way is to develop novel methods to take abundant unstructured data and turn it into structured data. And that's what I'm excited about. Okay, so Imageomics is the solution. And so images, like I said, are one of the most abundant data types. We have museum scans, lab micrographs, camera trap images, citizen science photos, even social media posts. But right now, most of these images exist really as raw pixels, right? If you want to know what trait is expressed in the photo or which species is pictured, that's a manual process. You have to annotate that by hand. And this is not going to scale with the abundance of images, right? And we're generating new images every day. And so that's what Imageomics is supposed to help us with. We're going to use artificial intelligence, computer vision techniques to systematically extract structured knowledge from images. So this might be the species identifications, morphological trait measurements, behaviors, environmental contexts. And then we want to integrate these insights in their structured form into the broader curated ecosystem. So just as genomics is the quantitative study of genes and proteomics is the quantitative study of proteins, Imageomics is the quantitative study of images. And so if I think for the biocuration community, this is the logical next step, is leveraging this new source of structured knowledge, images. Okay, so at its core, Imageomics, like I said, is about extracting quantitative structured data from images. And so we're going to achieve this at scale by developing novel automation methodology and leveraging domain-specific expertise where necessary. So I want to clarify one thing. Imageomics with a lowercase I is this new field of science that we hope to develop and share publicly. But the Imageomics Institute with an uppercase I is this formal group of researchers funded by the NSF here in the US, dedicated to the study of imageomics with a lowercase I. Okay, so there are a lot of projects within our institute. I'm going to give you a couple examples and we'll talk about some of them and then more of the macro level as well. So these are things like, measuring the wing lengths of beetles from images, identifying species using social media posts to do better species distribution modeling, re-identifying zebras from camera traps so we can understand migration patterns, counting plankton from underwater camera traps to quantify biomass in areas of conservation. And the idea is to do all of this from raw images rather than structured data already. And so the Institute aims to find common early stage challenges that are common across all these projects, and then share the technical innovations to reduce barriers to entry in this field. Okay, so let's talk about a concrete project, which is to measure morphological traits from beetles. And so they're a very, very diverse group. These actually right here are just carabids, which are just ground beetles. And they've got these very subtle morphological differences that make it very hard to identify them. And you can be an entire expert just in identifying beetles. And so a trait that we're very excited about is the width and the length of the elytra, which is this hard shell that covers the wings on a beetle's thorax. That's a pair of morphological traits that are extremely interesting at scale in a number of different subfields. So this could be like ecological entomology, climate change, evolutionary biology, conservation biology, and biodiversity informatics research would all benefit from having a large data set of images along with these structured elytra widths and lengths. So some of the questions we want to answer would be things like do beetle body sizes follow Bergmann's rule? And so are they larger in colder climates? Are warming trends on the planet causing meaningful shifts in morphology? Are beetle species converging into similar ecological pressures? And so I'm a computer science person, so sometimes you have to convince me that measuring 10,000 beetles would be interesting. But I am convinced, all right? And so I'm fully on board developing a large, multiple thousand, 10,000, 100,000 sample data set of elytra widths and lengths. Let's talk about how you could do that. So these are the two measurements we're interested in, the length and the width. And broadly, I think the overly simplistic view is take pictures, measure the traits, and then share the data. And I'm going to just ignore the hardship of taking these pictures because I've done it before. I'm sure everyone here has worked on stuff like that. I'll end up doing it again. I'm really not looking forward to it. And the challenges with sharing data, which again, I've shared large data sets before. I will end up doing it again. And again, it's hard. I don't really look forward to doing it. What I am excited about is step 2, measuring these traits. So I want to focus on this challenge of measuring these traits automatically, right? We can use software to measure these traits. These measurements were actually in Toro, which is like this image annotation software. And again, I'm going to continue to annotate traits manually, but I'm much more excited about building software to reduce that human effort in step 2 of measuring traits. Okay, and so anytime I repeat the same set of steps over and over again, I end up wanting to automate it. I'm a programmer. Right, this is like the golden idol of programmers everywhere, to see some task that's being repeated over and over again and then replace it with code. But just like Indiana Jones is about to find out here, that is not always as easy as it seems, right? Like it can be quite challenging to write reliable software to automate human effort, especially for things like annotating measurements on these images of beetles. Traditional programming is not a good fit, right? So machine learning, its ability to learn these like very arbitrary sort of heuristic-like algorithms from data without a specification of how to do so makes it a perfect fit for this kind of step. And like Alejandro said, AI is like, it's here, it seems like it's here to stay. But I think right now in the last couple of years is a really exciting time for imageomics. And so I want to explain why now is so exciting through the lens of this beetle project. Okay, so in 2018, just a few years ago, automating the extraction of standardized morphological measurements from beetles would have been like an enormous undertaking from a computational perspective. And so the approach, like the state-of-the-art approach a couple of years ago, would have required developing multiple bespoke custom algorithms. So first, we need a specialized algorithm to crop these collection drawer images into individual beetle photos. And then we would want probably another model to assess image quality and whether it's worth even annotating. Maybe there's some distortion that we can't get around. And then we would need another model to actually do the measurements, do the annotation, identify the landmarks or key points that make up the elytra width and length. And each of these steps would have required, likely thousands of manually labeled training samples, separate neural networks or models trained from scratch, extensive validation, testing, and then, ongoing updates as we integrate new sources of data. But now, we have these large generalist AI models. Maybe you've heard the term foundation models, but in general, these models are trained on millions and millions and billions and trillions of samples. But what they enable us to do is, develop really simple computational pipelines that don't need this like an entire thesis of work just to solve one problem. So, we're using grounding dyno to do this cropping from the specimen drawer photos into individual photos. That's like 97% accurate, and it took one of my lab mates like 20 minutes to set up. Then we're going to use Dyno V2 and train an SVM with like 30 labeled samples instead of 3,000. So there's another 100 times fold reduction in the number of samples we need to assess image quality. We're going to use Segment Anything V2 to do these actual measurements. And so I want to emphasize that, like right now, especially in the last couple of years, although the sort of scope of problems has not necessarily changed, the ease and the technical barrier to solving them is massively reduced. And so the question is not like, can we build this? Is it even possible to solve these problems? Everyone knows that it's possible. It's now what's fastest, what's easiest, what's going to be most reliable. And so, again, if you'd had this sort of interdisciplinary collaboration between entomologists and machine learning researchers, even just a couple of years ago, it would have been about, is it possible? And now, like in January, we started this project and there was no one raised the question of, oh, that's impossible. That's a thesis worth of work. It was like, well, when do you want this done by? September, or like is June when it needs to be done? And so this democratization of advanced AI capabilities, I think, is especially significant for biocuration. So small teams with limited resources can now build these sophisticated image analysis pipelines by leveraging these large generalist models, these foundation models. So the technical barriers have been significantly lowered. And maybe personally, I'm motivated to say it's still very hard. You still need me. You need to be experienced in Python, PyTorch, there's CUDA, you have these weird bugs that don't show up in typical software. You need a $10,000 computer that's going to draw more power than your washing machine, right? It's still hard, but it's fundamentally possible. And now it's just a question of how you want to solve it. Okay, so I'm gonna talk a little bit more about the beetle example, right? Now that we're gonna process tens of thousands of samples, we're gonna extract these standardized measurements that are gonna be mapped to ontology terms. And so this is transformative. When I talk to entomologists about designing these sorts of like trait-based ecology studies, I find that they often operate from a mindset of scarcity. And so we'll take whatever data we can get and we'll deal with limited sample sizes using statistical tricks in order to prove statistical power or find statistically meaningful insights. And what we're discovering is that through Imageomics and AI and these foundation models is that you can really flip that on its head and talk about operating from a mindset of abundance. And so instead of things like, oh, elytra length is a proxy for body size, and we're pretty convinced of that. And so we use that in correlation with things like temperature and latitude to measure, does body size correlate with temperature? You can just also measure the body size. Like it doesn't have to be a one or the other. Let's measure the elytra width, the length, and the body size. Maybe that's not a good proxy. And again, so now when you operate from the abundance or from the mindset of abundance, the kinds of questions you can ask change. You can challenge assumptions that you've been operating on for years. And so it really opens the door to like large scale trait analysis, the kind that can answer previously unanswerable questions about evolution, adaptation, and biodiversity patterns. Okay, so I could discuss one of the many other projects, right, like Zebra ID or counting plankton, but If we did these sort of details over and over again, we would start to notice this trend. And this is the use of these large generalist foundation models that produce this mathematically structured representation of images at various stages and various resolutions. So if you're a machine learning researcher, you basically spend all of your time thinking about the properties of these representations and how we can exploit these representations to do interesting things like species classification. And so I'm not sure about everyone's background, but I want to make sure we all have, at least one point of intuition about these representations. So we're going to do a little math and talk about vector spaces or embedding spaces. So the core idea about these models is that they're organizing images. Right, I focus on images, but we could talk about language or like genomic models. But broadly, we're organizing images in space. Okay, so here's a 2D space, right? The X and Y axis, I've put some points on this space, and then I've just colored them and given them shapes based on how close they are together. And so the core insight with embedding spaces is that points close together in our embedding space should also be close together or sort of similar in the real world as well. Okay, so here's an example. I have a bunch of pictures of dog breeds. And so now our points represent dog breeds, and similar breeds are closer together in our embedding space. So if we had, instead of an image model, if we had a dog breed model, you could give it as input a dog breed, and then it would give you a 2D point in space. And similar breeds would end up closer together in space. So English Mastiffs and Bull Mastiffs are together in that top right, and then Labradors and Poodles are off separate on their own. Okay, it doesn't have to be just dogs. We could talk about movies, right? Superhero movies are in the top right now. Star Wars is at the bottom. These musical movies are in the top left. If I'd included Star Trek, it'd be down there with Star Wars, right? Like sci-fi. If we had Superman movies in the model, maybe they would show, or likely they would show up next to the superhero movies, but probably closer to the Batman movies than the Spider-Man movies. Okay, one more example. Publicly traded companies, right? So I've got some consumer product companies like P&G and Unilever. We've got tech companies in the upper right, and then like credit card and banks down the bottom. So you can think about where you would put maybe a Wall Street firm like Goldman Sachs. Car companies don't really fit in either one of these clusters, so maybe they'd form their own cluster down in the bottom left. And so the key idea that everyone should internalize, hopefully, is that you can organize ideas, concepts, kind of whatever thing you want in these embedding spaces in a semantically meaningful way, in a way that reflects your understanding of the real world. So, okay, why is that useful? I think one like super obvious example here is that if I asked you to do some competitor analysis on Amazon, and you had never heard of Amazon for some reason, you could use your embedding space and then pick out the companies that are closest to Amazon in your embedding space as likely competitors. And so there's a lot of things you can do. That's just like one example of how you can use embedding spaces, assuming that you have a good embedding space. Okay, having a good embedding space. How do we actually make these embedding spaces? We want to talk about what's the best way to organize different concepts, what patterns are important to include. And for many topics, these might not have an obvious answer, right? You could think of lots of different ways to organize movies when they were released, what genre, director, actors, reviews. There's lots of patterns without necessarily an obvious answer. In biology, however, we do have at least one obvious answer, which is the Linnaean taxonomy, right? This genus species organization. We could say that two species in the same genus should be more similar, closer together in our embedding space than two species in different genera. Okay, and so to summarize sort of like the math, and then we'll stop talking about embedding spaces and vector spaces, is that we're going to use large generalist models for lots of different tasks within the field of Imageomics. And embedding spaces are a common way to organize information across different contexts. And the Linnaean taxonomy offers a natural organization, a natural way to talk about how concepts should be organized in our embedding space. And so it'd be really great if we had a large generalist model that took images of living organisms and produced an embedding space that respected the decades and centuries of organization done by biologists to organize living organisms. And so that's exactly what we did, right? As the Imageomics Institute, we knew that for an individual project, it might not be relevant to develop such a model. But because we're an institute and we're seeing the common challenges across many projects, we knew we could dedicate the resources to building a model that solve these problems across multiple projects. And so the model we have is called Bioclip. Probably you guys are not familiar with CVPR, but it's a big machine learning conference. And Bioclip was widely recognized. It won Best Student Paper last summer. And so we're really excited that the machine learning community is excited about Imageomics and these problems of representing images as structured data. So I'm going to talk about how to make Bioclip, but again, Bioclip is just a model that takes images of living organisms and dumps them into an embedding space in a way that respects the structure present in the tree of life. So to train these kinds of models, you need a lot of GPUs, so a lot of compute power. I'm very lucky. The Imageomics Institute provides a lot of resources to me as a student. You need some technical expertise. Mostly I have great lab mates to help me with that. And then large, clean, diverse data. And I'm sure everyone here is equally excited about large, clean, and diverse data as I am. And I'm sure you know this is a big challenge, right? This is every machine learning researcher's dream, but in practice, it ends up being just a dream. It's a huge challenge. So because of the big challenge, we made a big deal about it in our paper. And so we published this large data set called Tree of Life 10 million. It's 10 million images of living organisms from 3 different sources, unified the taxonomies, and then we made it easily available to other machine learning researchers. This is often a huge barrier to getting, developing new models in a particular field for machine learning research is making data easily available. And so this was a big effort for us. It's up on Hugging Face, which is a very popular data sharing platform for machine learning research. And so I want to talk about specifically the large and diverse parts of Tree of Life. Okay, so before Tree of Life, before our contribution last summer, there's existing work. INAT 2021 is a very popular one. It had nearly 2.7 million images. Bioscan 1 million is a million images of beetles. Sorry, not beetles, but all insects. But we went out and we leveraged this great public resource, encyclopedia of life.org, which is this website that curates images of living organisms. And we collaborated with them and downloaded 6.6 million images. So we roughly tripled, right? We went from 3.7 million images between INAT and Bioscan to about 10 million with EOL. And large is great. What I'm much more excited about is the diversity component. So prior work, INAT and Bioscan were in the tens of thousands of different taxa, different species, and we added 448,000. So that's nearly 40 times or 45 times more species. And we do a lot of ablations in our paper, and we demonstrate this diversity is actually the reason Bioclip is such a strong model. It organizes things much better than existing work. And so, again, data is the primary driver of model quality, and we're going to talk more about that later. Okay, so what actually can you do with Bioclip? Now that you've organized images of living organisms into this nice structured space, who cares? So I'm going to talk about 2 very quick projects. One is you can develop efficient classifiers. A lab mate of mine, Michelle Ramirez at Ohio State, is interested in doing better species distribution modeling, and she's using citizen science and social media posts that have been tagged with geographic locations to develop better models. Species models, not machine learning models. And so Flickr is this, image hosting website, and people will tag images. And so if you look up all the images tagged coyote, you will mostly get coyotes, except for those 2 birds down on the bottom right. That's okay. Bioclip is really good at this. If you organize things in your embedding space, it's very easy to draw a line in that space or a plane and say everything above this line is a coyote and everything below this line is not a coyote. It was kind of hand wavy, but I promise it's very, very simple compared to before Bioclip. And so Michelle is filtering these citizen science photos, these social media posts with 20 labeled samples, and she's producing much more accurate species distribution models of coyotes across North America. The other thing you can do that I really didn't appreciate until having the opportunity to go out into the field is iterating on study design in real time. And so Bioclip has this demo online where you can upload a picture and then ask it to give you a probability distribution over all species, nearly all 450,000 different species in the training data. And what's amazing is that, if you have a network connection, this is on your phone, this is on your laptop. And so you can test 10 to 20 to 30 images, in 30 minutes. and get a sense for where the state-of-the-art models are in terms of what they can and can't achieve. And then you can update your study design in the field sort of on a day-by-day basis. So more concretely, I was in Hawaii in January developing some phenotype studies where they wanted to understand the number of flowers over time on plants. And we were using BioClip to detect whether it was flowering or whether there was a bird in frame or not. And watching my biology collaborators use the demo to update their experimental design of how many cameras we needed, how many like replicates we needed was phenomenal, because you watch the study go from something that could have collected basically no useful data and watch it update in real time until they had figured out, okay, we need 20 cameras and I need, this kind of motion trap, and then I'll be able to expect this accuracy. And so like the zero shot nature, the very data efficient nature and demos associated with these foundation models is broadly very, very useful to biologists. And I didn't appreciate that until going out into the field in January. Okay, so I want to understand BioClip. This is like machine learning interpretability. And I think there's two main parts you can look at: the data, so that's tree of life, or the model itself. And broadly, people will tell you that you should look at the data first, right? So I don't know if you've heard this phrase, garbage in, garbage out. It shows up a lot when we're talking about training neural networks. This idea of data quality being the primary driver of model quality. And, I talked about the diversity in tree of life leading to improved model quality. So I'm already on the train of like, you need the best, highest quality data possible if you want to get a good model. Okay, so I don't know if you guys have seen this comic, but machine learning is just this big pile of linear algebra, and you sort of just pour your data in, and then you end up with answers. And so that's why we spend a lot of time, because the linear algebra can be hard, and a concrete way to improve your model quality is to improve your data quality. But once you sort of get past that, you might ask, what's actually in our models? What's going on inside that big pile of linear algebra? So this is the field, like I said, of machine learning interpretability. And so I think one analogy is that just like psychology tries to understand human brains and like our minds, machine learning interpretability is trying to understand these big piles of linear algebra, these huge, massive multi-parameter models. And so I think machine learning interpretability has focused primarily on interpreting predictions. And so these are techniques like saliency maps or GradCam, attention heat maps, attention visualization. It's all about when a model says, this is a blue jay or this is a coyote, you say, okay, what about this particular image makes you think it's a coyote or a blue jay? And the Imageomics Institute has contributed to this body of work. We have papers like Inter, FinerCam, PromptCam, all of which I'm excited to talk about, but after this keynote. Today, I want to talk about a different technique that's all about interpreting the model itself. And so instead of focusing on an individual prediction or trying to interpret a set of predictions, a sample of predictions, and then using that to say something about the model, we're developing new interpretability techniques to directly interpret the model and try and understand what the model knows and what the model doesn't know. So we talked about embedding spaces as a way to organize information. And I want to dive even deeper into why these spaces are important and how we can better understand what's going on inside them. So these spaces, because of their organizational structure and their high dimensional structure, right? I used 2D up here. In practice, BioClip is representing things in 768 dimensional space, but they capture these very rich information about organisms. So this is like taxonomic relationships, obviously morphological traits, ecological niches, and more. And part of the problem is that and more is really loaded. We don't actually know what pattern, what structure is being represented by our models. So that's what we want to learn. So the challenge is, while we know that there's structure in these spaces, it's difficult to extract it. Models are learning to organize our data, organize our images in ways that are mathematically optimal, but not necessarily intuitive to humans. So there's actually a whole line of work that goes the other way and says, okay, let's organize data in a way that's intuitive to humans, but not necessarily mathematically optimal. I think having a mathematically optimal organization is better. We'll just figure out how to interpret it afterwards. And so because models don't learn like us, they learn these multiple organizational structures simultaneously. It can be really challenging to decode what's going on inside them. Okay, So sparse autoencoders are this new technique in the field of machine learning interpretability that I'm very, very excited about. They were originally developed for large language models, and the Imageomics Institute is sort of pioneering the use of them for vision models as well. And they decompose complex embedding spaces into more interpretable components. And so the sparse part of sparse autoencoders is super, super important. It means we're forcing the model to explain its representations in as few features as possible, which tends to lead to these more human interpretable results. And so in practice, when we apply these sorts of models to BioClip, we get these really cool patterns that we didn't expect to see. And so they're enabling sort of scientific discovery or discovery within the models. And then we want to leverage the discovery of what's going on inside the models to enable scientific discovery in the real world. Okay. So sparse autoencoders are converting these low dimensional vectors, low dimension 768 dimensional vectors, which is just a list of 768 numbers, into actually much bigger vectors, much, much bigger, like 24,000 dimensional vectors. And so while that seems bad, we're making things more complex. Now we're representing images. Instead of image goes into BioClip, BioClip produces a 768 dimensional vector. We turn it into 24,000. The neat bit is that most of those 24,000 numbers are 0. So like more than 99% of them are all 0. And in fact, fewer than 768 of them are 0. So we're sort of compressing our information now into a sense of like position and non-zero-ness. So the amount of meaningful information has gotten smaller, which makes it much easier to interpret. But we don't know what each position in this long list of 24,000 numbers means. Okay, so let's show some pictures again. This is a big graphic, but in the middle we have our, I can use the pointer. Excellent. In the middle, we have our 24,000 number list, and most of those are 0 for most images. But what I might be interested in is understanding what's going on in this 1,000th position in the list. So when an image produces a vector, produces a 24,000 dimensional list with a non-zero element at position 1,000, like what does that actually mean? And so the way we do this right now is you filter through a bunch of images of living organisms and take all of the images that produce non-zero values at position 1,000. So you might end up with things like striped beetle thoraxes. And so these patches, these colors are just ways to highlight what specific part of the image is contributing to that non-zero-ness. Another example might be like position 2012, and we end up with moth antenna as being the parts of the image that are making this vector non-zero at 2012. What happens is that because for most images, 99% of those numbers are 0, it's very unlikely for an image to be non-zero both at moth antenna and striped beetle thoraxes. So you get these nice, very precise representations, and we call them disentangled representations. And this leads to a lot of new possibilities, and so I'm going to talk about just a couple examples. But I think this is very early stage work, and I'm really excited to see how we can leverage this for more structured knowledge in the future. So one is just better search. Right now, if you want to find images of, say, moth antenna, but more broadly, like maybe a bird feeding in a flower, you have to use text to describe that process, right? This is sort of just like Google image search. And the ability to link text and images is quite challenging. This is like the grounding problem in machine learning research. It's ongoing. What's exciting about sparse autoencoders is that you can give your search engine examples of what you want to see and say these specific parts of the image are what I'm looking for. And then you can use this very sparse representation to find images that also fire on, say, position 2050 or whatever, position 3000. So now you can discover unexpected patterns, right? Maybe there are certain coloration patterns that are consistently co-occurring with specific behaviors. Maybe morphological traits are predicting particular habitats. And so these connections and these co-occurrences that we can use with this nice structured sparse representation might represent important biological insights that were hiding in plain sight in our image collections that we didn't know because we were using text instead of this structured representation. We might have better integration, right? So once we have these more semantic embedding spaces, we think they're going to be far more integratable with existing knowledge graphs and ontologies. And so hopefully we'll be able to link visual data with more structured knowledge graphs, like I said, or other curated data sets in a way that was previously impossible before we started to dig into what's going on inside these models. And so the way we think about sparse autoencoders is that they're helping us decode this language, the structure that our models have learned. And we want to transform these abstract embedding spaces into biologically meaningful concepts, again, hopefully to advance scientific discovery. Okay, so I've talked about three sort of major projects from the micro scale up into this, the micro scale with beetles is a very specific project, and then Bioclip, which was a method to help all Imageomics projects, and then sparse autoencoders, which is now looking at Bioclip and trying to understand deeper what's going on inside that model. And so despite, the promise of all the work we're doing, there are, of course, still open questions. I'm very lucky, it gives me a job. Right, I think the number one that we see in machine learning research, and I'm sure that you're all aware of, is data bias. And so existing work before Tree of Life, right, only had 10,000 species. These are the most common species, maybe, well-lit museum specimens or species that are popular in North America in areas, in wealthy areas where you have a proliferation of like mobile phones and digital cameras. And so we're very excited about developing more diverse training sets, both geographically and temporally, and again, especially for underrepresented taxa. And then there's this quote in computer science: there are only two hard problems in computer science, naming things and cache invalidation. This naming things really scares me. When you talk to a biologist about what is the definition of a trait, right? And sort of a trait represents like the fundamental unit of structured knowledge about living organisms. If you ask 5 biologists what is a trait, you'll get 5 different definitions. And so naming traits seems to be very, very challenging. Agreeing on what is a trait versus what isn't a trait seems to be hard. We have changing names, right? I'm sure everyone's knowledge graphs are being constantly updated, and we're handling new morphological concepts, new behavioral concepts, new scientific discoveries. So I'm actually very excited to learn from you all about how you deal with naming things and like updating the names we use to refer to different concepts. And then finally, computational framing, I find very, very challenging. This is taking problems that, Chuck and Alfie did with a bunch of notes, right? Their latent space of notes about the idea of evolution. How do you turn that into a problem that is amenable to our algorithms, our big piles of linear algebra? Large language models might help in that we can turn this unstructured textual representation into something more structured. I don't think that's the solution to everything. And so thinking about how we can turn these broad fuzzy concepts into mathematically precise definitions that are amenable to optimization is something that seems very challenging, but I'm very excited about in the future. Okay, and so I'm gonna summarize here a little early. Images are a still untapped resource for understanding the secrets of life at scale. And this might be micro level morphological traits or macro level diversity patterns. So Imageomics and the Imageomics Institute is all about using knowledge guided AI to produce structured data from these unstructured images. And I want to emphasize this is not about replacing curators, right? It's about how we can handle and building tools to handle exponentially larger data sets, and then leveraging tools to drive and discover insights that manual methods alone might miss, or that existing methods are going to miss. And so I'm very excited about the future of ontology-driven image data, merging with textual descriptions and genomic data, and then how we can leverage all these different modalities to fuel scientific discovery and deeper scientific understanding. So thank you all for your attention, and I'm happy to take any questions.
And there's 2 mics down here. I think Steph now is getting a mic today. There's 2 mics, so you can come down to a mic and ask a question or get Jay.
Great talk. Yeah. Okay. When you think about the future applications, where are you dreaming this is going to go?
I'm really excited about sparse autoencoders. I'm gonna flip back to this page with all these examples. And so, when we talk to biologists, one of the big problems is, well, your machine learning model is great for distinguishing between tigers and lions, but it'll never be able to distinguish between these 9 different rodent species that look very similar, or these butterfly mimics, these butterfly species that grow to look like each other. So I'm very excited about using these models to find distinguishing traits that humans can't find, right? And so finding the subtle patterns in large scale data in a way that is scientifically meaningful. We talk about trait discovery all the time at the Imageomics Institute. Yeah.
Excellent talk. Thank you so much. I'm struck by all of it, but what I see is a lot of images that all look the same, that are acquired the same way. When we look at, let's say, microscopy, for example, we have many different ways of imaging things. And so I wonder, how do you guys imagine BioClip handling things like night vision images or thermal images? That's something you've been thinking about.
Yeah, definitely. So I'm going to give you the cop-out answer, which is sort of in future work we trust. But no, this is version 1, and the idea is that we're going to integrate more data sets. So the next version is going to have 20 times as many images, so over 200 million images that we're training on, and we're deliberately integrating data sets with night vision, thermal imagery, non-sort of RGB. But the idea is that if you learn to understand what's going on in pictures like this, a lot of the model's sort of understanding of the world can be applied to black and white images as well, right? And the same physical structures are present in the real world.
Anybody has any other question?
Thanks. That was an incredibly coherent talk. Thank you very much. One question I would have would be going back to the garbage in, garbage out problem. How, I mean, you did get, how do you envision getting massive, large enough, I'd put it this way, amount of well-curated data for your training? Because here you're applying basically kind of a crowdsourcing approach, but this is a crowdsourcing approach that is relatively, say, easy to acquire because most people can tell the difference between a dragonfly and a beetle. But if we go back to the question that was asked here about microscopy images, suddenly, not a lot of people can distinguish between the zebrafish notochord and stem, right? So how do you see that happening?
Definitely, that is a great question. To me, the most, there's 2 pieces to that. One is I think machine learning is exceptional at deriving signal from noise with enough scale. And so the idea that 10%, 20% of your annotations can be wrong, but the model will still sort of learn the underlying structure. Definitely cleaner is better, but I do think machine learning is a powerful technique for dealing with noisy data. The other piece of that is self-supervised learning. So one of the things about Bioclip is that we took this existing organizational structure of the tree of life and sort of superimposed it into the embedding space that Bioclip learned. Self-supervised learning is much more about letting the model discover its own organizational structure without any superimposed structure. And so rather than give it an image, predict the species, it's given half an image, predict the other half of the pixels. And so the only way to sort of, say, given the left half of the image, predict the other half is to have this understanding of what's going on in the physical world. And there's lots of ongoing work about developing self-supervised techniques. They seem to be very powerful for learning from unlabeled data at scale. And so hopefully we can leverage that for Imageomics as well.
Do we have any other question from the others? No? Okay, you can catch some outside as well. I think we can move forward to the coffee break, okay? We are meeting here at 10:55, okay?
I can introduce myself.
And then, yeah, you have that, and the pointer, right, is at
It's
the
on
top
the
of
top,
this.
yeah,
I
yeah.
saw him use it, Sam. So.
Yeah.
All right, I'm gonna get water here. All right, guys, we're gonna start. Who will introduce himself?
Hello, welcome back. So I'm introducing myself. I'm Chris Hunter, on the slides up there. So I think we're pretty much all back in the room now, so it's great to see you. So thanks to the organisers for giving us this opportunity to speak about GigaDB and GigaScience. I'm going to try and give you a little bit of an intro to what GigaDB is and what I do and my team does. The title, we might get around to a little bit of, of encouraging the citation of data, but more about how you reference data from our data set. So it's sort of slightly misleading title maybe, but you can ignore the content slide because I didn't update that. So GigaScience Press is the overarching body of what we are. So GigaScience Press is a publisher, as you'd expect. And we have two journals, one called GigaScience and one called Gigabyte. GigaScience has been going for like 13 years now. Gigabyte was launched in 2022. And the other unique selling factor, if you like, about GigaScience is the fact that we have this database called GigaDB. It's a repository for hosting any data that will make either of these two journals, any of the articles in either of these two journals, as fair and as open and as accessible as they possibly can be. So I run the database. I have Bastian here, who's one of the curators in my team. So there's two of us here if you want to come and speak to any of us. And essentially what we do is we look at every paper that's going to be published before it actually gets published with these journals. And we go through methods and the results and make sure that all of the data is available. That's sort of basically covering what's just been said and make sure that all the data is organized and that people can access it. And we're now starting to move towards encouraging the proper citation. So there's now a lot more rules around how authors should cite this data to make it to get the credit for it. So when you actually publish your data set, it's not just lying around somewhere and anybody can use it. They actually have a way to cite it in a manuscript, the same as they do for other manuscripts. That's how we're hoping to make things more countable, more give you the credit for producing these things in the first place. We follow the FAIR principles, so we do try and make everything as accessible as possible and reusable and interoperable. So we encourage all the authors to make sure they're using a format that's appropriate for their data and that is maintainable. I mean, we go to the extent of not hosting Excel files when we host tabular data. We make sure it's in CSV or TSV because it's more machine readable. Okay, Excel is probably a bad example because pretty much everything can handle Excel, but you get the meaning. We try to encourage things that are open formats rather than proprietary formats. All of our GigaDB datasets are given DOIs, which is part of the way that you can make these things sustainable. So it's a persistent identifier. So that DOI, even if we move our stuff on our servers and it has a different URL at some point because the files will be on different places, the DOI will be updated and it will always be the same regardless of where we put our files. So just a little bit about the structure of GigaDB. We have there's sort of three aspects to it really. We've got the website, which is the sort of front end bit that people will come to and use and see. But we also have, because we're all open, the code that builds that website is all on GitHub. So if anybody wants to assist in development of GigaDB or has issues that they want to raise, there's GitHub tickets there. You can put in a Git ticket, say, can you do this? And with your browser, then there's that option. So then the back end is all a PSQL database. So the actual metadata that's hosted and shown on that website is all in a database. And then there's the cloud server, which is for the data files themselves. So they're sort of separate parts of the infrastructure that we have to look after. So we have a development team as well as a team of curators. So just a bit about the sort of workflow that we go through. So the author is obviously center. That's the important part. The person who's writing all this thing, these papers or creating this data, they're the ones that drive this whole process. And they will submit us a manuscript to either GigaScience or Gigabyte. And then the editorial team will do their bit. They will assess it for whether it's within scope, whether it matches the criteria that they have for publishing papers. If it does, they will then pass it to my team, and we will look at it with a critical eye to say, does a peer reviewer have everything they need to assess this manuscript? So that's kind of the basic question that we ask. And if not, we go back to the author and say, where's this? Where's that? You know, put it here. Do this. You know, we make authors jump through hoops before it even goes to peer review. so that the peer reviewers can actually reproduce the paper if they want to. I don't know how many of them actually do, but that's another question. So once it's all available, we've confirmed it's available, it then gets past the next step, it goes back to the editorial team. They will do their bit where they go off and ask 46 reviewers if they want to review this paper and get one person to say yes, or whatever they have to do. It's sort of beyond me what editors do. And eventually it comes to the point where the editorial team can make a decision on whether they need to revise the paper, in which case they communicate with the authors again and do revisions. And sometimes that can mean revisions to the data as well, because if they have to do more experiments or they have to bring in extra data. So that sort of communication happens. And then once it's ready to be accepted, they will do a provisional accept to the authors and then pass it back to my team in the GigaDB, where the curators will look at it again and make sure that not only is it accessible to peer reviewers, it is now openly accessible. So we'll make sure that all of the data is published. So the sequencing data in the ISDC, if it was given a private reviewer access, we make sure that it's now published and open so everybody can get at it. And similarly, we publish the GigaDB dataset at that point so that the authors can then put that DOI and that citation of the dataset into the manuscript. So that's sort of going full circle, then eventually the editorial team will publish it on the website, in the journal, and it will get indexed in all the relevant places. So what we do is curate everything. So I think I've kind of gone through this as I've been speaking, but essentially we sort of break it down to 3 levels. So there's the sort of overarching data set part where we link out to all the different repositories, and I'll show you a few of those in a moment. And we have the sort of more granular level. We spend quite a lot of time looking at the samples. So because we're all across the life sciences, a lot of the data sets and the manuscripts we publish are to do with sampling and sequencing and those sorts of things. So our curation team actually look at the sample metadata. We encourage authors to put it into whichever repository they've also put the data in. So if it's sequencing data, the ISDC, or Metabolomics data in one of the Metabolomics databases. We can't force them to do that, but we do encourage them and assist them in using the correct terminology to do that. But we also put that information in GigaDB, so if you need more sample information, it is at least indexed in GigaDB. And that includes, well, I don't know if you saw the GSC slide up there, so I'm a member of the GSC consortium, and we make sure we use the standardized terminology for the attributes associated with the samples as well. So we've got a link across there to another sort of standardized way of producing this metadata. Similarly with the files I've already mentioned, we make sure that the formats are all there. Importantly, one thing that we do is have descriptions on every file. So every file that is in GigaDB, okay, I'll caveat that. The first 3 or 4 years we were running, we didn't insist on this, so you will find some files in GigaDB that don't have descriptions, but not many. So every file now has a description that tells people what is actually the content of that file, because it's not always obvious from a file name, especially if they've got sample1.csv. Yeah, the names of files don't necessarily help much. So we make sure that the file descriptions are there in the database as well, along with the formats and a type. So our types we use just a set of different types so we can classify the different types of data that's within the database. All of the files get MD5 checksums so that you can actually download and check that you've got exactly what was provided to us in the first place. And obviously make sure that everything is as open as it can be. Just want a quick run through of what a data set looks like. If you want to follow it, it's that DOI number. It's an imaging data set, actually, which is quite apt after the morning talk. So this is sort of the top half of what our GigaDB dataset page looks like. The types I mentioned there, so this is an imaging type, and then you've got the various different sections. So this is sort of the dataset level metadata, if you like, where we have the title, we have the author list and the citation of how you would cite that within your manuscript. The DOI number, obviously, because it needs a position identifier. The descriptions is something that we're looking at the moment. At the moment, the description is essentially a copy of the paper abstract, which isn't entirely relevant to the data set. So we're looking at ways that we can perhaps use AI to define more describe what the data set is rather than the abstract of the paper, which sometimes isn't entirely relevant to the data set. And then obviously we do have a contact submitter, and we have the opportunity for other authors to claim ownership as well. So at the moment, it's only the submitting author that is in GigaDB as a contactable person. Whereas if you're one of these other authors and want to have your name associated with it, you can also claim it. A bunch of keywords, which are author input, and then citation sort of statistics, if you like. You can sort of see how many times this DOI has been shown up in Google Scholar or EuroPMC. I think Dimension, I think we've actually removed Dimensions now because they don't offer it for free anymore. This is the second half, so lower down the page, if you scroll down, you'll see this sort of section. Again, if it's already, so because we publish the data set first, we have to add in the manuscript links afterwards because the manuscript hasn't been published at the point we publish the data set. So usually that gets added in afterwards. So the peer-reviewed manuscript publication is linked there, and you can see how that's cited. And then there's these additional information links. In this case, there's only 2, but there could be a whole list of them depending on which data set is and what sort of data is associated with it. And they're just links out. So at the moment, it's just a URL, which isn't helpful. So we're building in the way to add descriptions to these, but bespoke, so that an author can actually say, this is the link to X, and actually describe what it is. Hopefully that'll make it more useful to people as well moving forward. And then at the bottom, we have these sort of tabs. So you saw the previous slide has got the sample information. So this is just what I was saying about all of the metadata we add into each of the samples. Similarly, with the files tab, we've got name, its description, and the type, the format, the size, and its MD5 checksum. We also add in other metadata. So in this case, we've got voxel size. So it's author-led. If people want to add more information to their files, they can. And then this is just a little display of all the different things that we kind of link out to and what our curators have to know a little bit about. So it just gives you an idea of the fact that there's so many different things out there that data can include that we have to try and keep on top of. So if you ever come to us with a submission for something and we don't know what you're talking about because it's not one of these, then yeah, bear with us. There's a lot of things that we need to keep a track of. But why go to all that effort? I think I've been going through as it is. It's make it transparent and reproducible, and hopefully machine ready at some point. And we're trying to make everything as accessible to machines. So all of the metadata is not only visible to a human on the website, it's going in the back end under schema.org. So we can actually have it scrapable and searchable in that respect. And it increases the exposure of the related articles. So just a couple of things on what we're working on. So I've mentioned a couple as we've gone through. We want to build, so at the moment, the curators are doing a lot of this work in sort of hand-holding and doing things for the author. We want to try and automate that process. Automate, that's not the right word. We want to try and assist the authors in doing it themselves rather than having to tell them to do it. And I can see the clock's gone red, so I better hurry up. And that's it, actually, just an acknowledgement. Thank you all for listening, and thank you to the authors that have given us the opportunity to produce this sort of work, and all the open repositories that we work with, certainly on that previous slide. If you want to join our main list, it's on gigadb.org, or come and find me and I can give you a card or whatever. That's the bunch of us that are here. So you know, myself and Bastian are here if you want to speak to us about anything. But post number 7, we've got a bit more information. And then I just want to leave you with the Genomic Standards Consortium. I will be talking about it on Wednesday, but it's a bit late by then, so I'll put up the post now. We've got a meeting coming up in July in Cambridge, UK. So if anybody's interested in that, again, the QR code, or come and see me, all in. Thank you.
Chris, for your talk. We have 2 minutes, so if anyone wants to ask a quiz.
Yeah, excellent talk, and we really appreciate that you are looking at the data and making sure it is well curated and available when the publication coming out. That is very helpful. And as you pointed out, the abstract may not be the actual good description of the data, and you already recognized it. And sometimes this data can be published multiple times, so tying with a single abstract, it can be maybe a little problematic. So I was just wondering, like, since you have already the sample information and also the kind of experiment that is done to generate this data, so why can't that information, are you looking at the possibility of using that information to describe the data rather than the abstract itself?
Yeah, essentially that's what we want to do. We want to use the data set to be able to write a more appropriate abstract or description of the data set. We could ask authors to do it, but we ask authors to do a lot at the moment. So it's kind of that's one step too far when we can take a good enough proxy from the manuscript at the moment. So yeah, the idea, or at least my idea at the moment that we haven't looked into is literally just getting an AI to go across it and suggesting a data set description that the authors can then say, yeah, this looks all right, or no, it should change it to this. So it's yeah, essentially using the stuff that's we've taken the effort to curate should be able to create the data set description easily enough.
Thank you.
Sorry, you can catch Chris.
Thank you.
Again. Valerie?
Okay, my name is Alan Barron. I work for the Human Disease Ontology. I'm going to talk to you today about our efforts to translate that ontology into various languages, mostly focusing on the first language, which is Spanish, and how we're sort of approaching this. So the Human Disease Ontology is a global core biodata resource. It was established more than 22 years ago in 2003, and with the goal to provide a unified etiology-based classification of all human diseases, including diseases caused by infectious organisms, by genetic mutations or modifications, by social and environmental factors. It also includes, our goal is to include all common and rare diseases, and we cross-reference or map to various important medical vocabularies, including OMIM and UMLS. Furthermore, as an ontology, we semantically integrate a number of disease-related factors, including anatomy, inheritance, phenotype, and various other things that you can see on this list. So we chose Spanish first, and why Spanish? And there's a couple of reasons for that. But first, here in the United States, the Hispanic population is the largest minority, and it is growing quickly. So if beyond that, Spanish is the second most spoken native language in the world. So there's incentive there to reach out to this population. The disease ontology is used globally. It's been incorporated into more than 400 biomedical resources and databases in more than 45 countries, including a number of Spanish countries, which you see here. It's also been, our website, diseaseontology.org, in just the last year and a half, has been accessed from more than 122 different countries. And so there's a global reach there, but also Spanish speakers are always within the, no matter what time frame you look at, always within the top 5 number of our users. So we're not the first ontology to address translations. I think the most notable is the human phenotype ontology, which started maybe 10 years ago and trying to outsource translation to various communities. And this is a continued ongoing effort. And as part of their efforts, they've built some software that is useful for ontologies in terms of loading ontology translations, as well as managing changes to that, which they've titled Babylon. More recently, the mammalian phenotype ontology was translated by a group that wanted to have those in their native language of Japanese. But most OboFoundry ontologies still have not been translated in any way. So we're trying to join that group and make things more accessible, open, and fair to a broader community than just the English speakers. So we wanted to make sure that we had high quality translations, and our goal with this first approach in approaching these languages was in part to learn about the process and what kind of things might go wrong in translations. So we decided to start from the very best that we could, and we got the ontology, all the text in the ontology. So not just the labels, but also the definitions and the synonyms translated to Spanish by expert medical translators. We actually had the translation done in 3 groups. First, we asked them to translate our website text so that Spanish speakers could find and navigate our website and find the data they were interested in. Then we sent them a set of diseases that are all from the physical disorder branch so that we could sort of identify issues that might be available there. And then lastly, we send all the remaining diseases. So most of what I'm going to be talking to you today is not their translation because they're expert medical translators, but how do we find issues with their translation? Because even experts make mistakes, and we wanted to make sure that these were as high quality as possible. So what I'm going to talk to you mostly about today is how did we, as curators in the ontology, before loading these changes, verify and validate those translations? How did we find potential issues with them? And to do that, we sort of used a 3-step process, which I think you could use for pretty much anything. We used Google Translate and Google Sheets. We both forward and reverse translated. And I'll talk more about how that translation worked. And then after making translations separately with this machine learning model, we established a sort of code and for comparing the different translations and how they were. And then lastly, for those that didn't pass a certain cutoff or whatever, we would, or even those that did, we, it basically bin these things into different ways or of different depths, I guess, of review that were needed. So, and you'll see that on the next couple of slides. So we first had them translate the website data, and they did a good job of translating, which we hoped for and expected. We noticed that there were some things that kind of came up that we also probably should have expected, but really didn't. Like, they didn't do a great job of translating ontology related terms in our website. And that just emphasized to us the importance of domain expertise, which is part of the reason we were like, wanted to have medical translations, because these are all disease names. But we were able to fix those. Also, we noticed that they translated a lot of things that we thought might be more confusing by like the human disease ontology name and other various names that we had. So we untranslated those. And these lessons that we sort of learned were learned by Claudia, who's a native Spanish speaker. She did most of like the reviewing of the translations and things to find gotcha. She grew up in Spain, but also somewhat by myself. I spent 2 years in Guatemala, and I'm maybe not native, but I am pretty fluent in Spanish. So mostly we just realized, hey, we're gonna have to like find a way to review these things. We thought initially when we sent them, we thought, yeah, medical translation, we won't have to do anything, but we actually had to do more. So when we sent the first set of the data from the physical disorder branch, we discovered a number of common issues with the translations versus what we were expecting for the ontology. Some of them were stylistic. So they basically fell into sort of two categories. Some were character differences, some were word differences. Like, a case change between like the word syndrome is more of a style difference. But then when we talk about the order of wording, potentially how they format, like what words are involved and things like that, then it becomes more of a content. And it was challenging. So we devised a way to sort of compare these two translations between Google Translate and what we had. So we did this for both English, the English, original English, and then back translated English from the Spanish translation. And then we compared separately the Spanish translation with the Google Translate forward translation. And I'll show this in more detail on the next slide. But I just wanted to make you aware that we're using here like 3 translations, 1 by expert medical researchers, 1 and 2 by Google Translate. And this was just so that we could develop kind of a scoring algorithm and a comparison model for how to determine what the differences were. And the scoring that we put up is there, and I'll talk about that in a second. But I'm not going to go into exactly how that works. Okay, so to walk you through things, this is an example with just one label, which is Ross River fever. That's the original English. So the Spanish group that translated these things for us translated it into Spanish. And then using Google Translate, we back translated their Spanish to English and forward translated our original English into Spanish. And then we compared the two English together, which is this sort of orange line, to see sort of what the differences are. And you can see that this has a difference just in case. And then the Spanish, we compared those, and you can see here that the differences, they have all the same words once you remove something like stop words. So meaningless words like of, but these are obviously Spanish stop words and not English stop words. But you can see that these are essentially both pretty, like really good translations. You would consider them basically equivalent. And they both, and so what we did was we developed the scoring algorithm based on, or this equation based on these differences. And then between the English and the Spanish, we just averaged them and considered that a final score. And anything that had a final score of greater than 0.75, we considered to be extremely high quality and needing very little review. And anything below that, which I'll show you in a second, is not as much as you would think. or you would think, I guess, with an expert curator, but is not that much. Our goal was to limit how much we had to really spend time re-reviewing any Spanish, or like if we're outsourcing this in the future, how long do we want people who are outsourcing things to look at that? All right, so we had translated over 11,000 labels, 18,000 synonyms, and 9,000 definitions. And you can see sort of the plot on the right-hand side, sort of the tree map, most of those, more than 40%, at least with the labels and synonyms, are like exact matches across all the English and the Spanish. So they're super high quality. They're exactly the same, not even character changes. And beyond that, another 35 to 45% passed our cutoff filter, and we consider them extremely high quality. So most of the labels and synonyms were great, definitions not quite so much. We incorporated this all into our website with just a single click button. You click the English button and it converts itself to Spanish, and it converts both the whole website using the Python package Babel and the disease ontology searchable data, which is the data that we loaded into the ontology. We're still in the process of getting that data loaded, but right now we cover 64% of the ontology, including 86% of the labels, 76% of synonyms, and 13% of definitions. If you want to talk more about our other efforts to verify the disease ontology and open our data, or talk about, I'll be later at our poster, which is number 4 today with Claudia, and happy to discuss that or the API or anything else. This is the team that did our work, that works with us, including our clinician team, who reviews a lot of complex disease situations, and I'm happy to take any questions. Oh, and we do have currently a user survey for our website, so if you're a common user of diseaseontology.org, please use that QR code to answer our survey. Thanks.
Gracias, Alan. Is there any questions?
So there are a lot of regionalisms in Spanish, like there are in English. How do you resolve when the difference is correct in 2 different ways?
Yeah, you're right. There are, including like closer you get to the United States, you get more and more Spanglish. We don't have a way to resolve those sorts of things, and what's like we don't have a way to do that. I guess we like ultimately. I think ultimately. I mean, the answer is if it would be right. It just like to which population is it right? You know, like, and so I mean, I know we do in English sort of keep British synonyms sort of but we don't have, and we have a sort of automated way to create British synonyms for English, but we wouldn't be able to create something like that for every language. So we don't have a way to manage regionalism. That's a good question. That's a hard one too. Go ahead.
Hi, very interesting. Thank you. I was wondering, have you thought about providing a plugin or some sort of module where portals that actually host the data could benefit from your translation work that you've done?
I'm not sure I can, like a module for
like I imagine
using the disease ontology.
Yeah, so like where the actual data sets are annotated with the human disease ontology. Now, let's say they're in Spanish or English, and we want to get the actual like application.
Yeah.
Of people using the data in that language.
Yeah, that is an excellent question. It's something that came up. Like one thing we've really noticed is like there's sort of software and infrastructure for getting translations into ontologies, but and technically RDF and OWL, which all ontologies are built on, have like many other programmatic things, they use language tags, so you can easily like pull those things out. But to democratize language everywhere is hard, even across ontologies. Like even incorporating something we've been looking at is even incorporating human phenotype ontology, which has translations, into the disease ontology and producing a file that's all in Spanish. We're still working on that, and it's a great point, and that's the first thing that came up when I first noticed this. But we don't have anything yet, and I would love to do that.
Oh, there's a question online. Shade is asking, what format are your translations made available publicly? Do you plan on making them available in Babylon format? Sorry.
Yeah. So right now they're in a CSV, which is like basically what Babylon uses. So they will be compatible with Babylon. They'll also be in the ontology and on the website. So anybody who wants to, pretty much any way that we can make them accessible, we plan to, although with the caveat of, we do have a couple of like servers for searching the disease ontology, which we have not yet incorporated Spanish into, including our faceted search and currently our Sparkle server. Those are things we haven't yet incorporated into. But otherwise, everywhere we can, it is there.
Thank you very much, Alan.
Sure.
Good morning, good afternoon if you're online. First of all, I want to thank the organizers for all the work they put in to get this meeting starting. So thank you so much, and thank you for giving me the opportunity to tell you about our work. My name is Sabrina Toro. I am part of the Translational Integrative Sciences at the University of North Carolina at Chapel Hill. I am also part of the Monarch Initiative. And so what we do at the Monarch Initiative is getting data from everywhere we can and integrating it so that we can compare the data and create tools to support disease diagnosis and treatment discovery. So in this talk, I will focus on disease. We are staying in the disease world here. And the data that we get for diseases are really coming from a lot of different communities and a lot of different sources. So we needed a way to get that data in such a way that we could integrate it so that we knew if one disease from one source was the same as a disease of another source. To do that, we created the Mondo Disease Ontology, and we still use the Mondo Disease Ontology to integrate our disease data. So the Mondo Disease Ontology is an ontology that integrates all key medical and biomedical terminologies. It provides textual and logical definitions for disease concept. Mondo unifies disease classifications and reconciles mappings between all disease resources. The scope of Mondo is very broad. So we try to cover all type of diseases, going from cancers, rare disease, genetic disease. And we also are aiming to represent all diseases from all species. So we have both a disease branch, a human disease branch, and a non-human disease branch. We are available in all the ontology browsers. Mondo is open source. Everything is on GitHub. And we have monthly releases. The product that you can find for Mondo is all the ontology formats that you can think of. We also have a set of mappings in the SESAM format. And we have a lot of subsets, such as the rare disease subset. Our team is not very big, considering all the things that we do. We are a group of curators, semantic engineers. We also have some domain experts, but we have a very, very big community that includes clinicians, database and terminology experts, groups that do disease data annotations, patient advocacy groups. So we really brought together a big community, and we really rely on this community to help us sustain Mondo. So one of the things that the community does is basically telling us what needs to be changed. So we have a lot of requests for changes in the ontology, a lot of reports of inconsistency. Anything that somebody needs, we try to add it into Mondo in order to keep the information relevant, accurate, and also up to date with what is happening in the clinical world. And we have seen that over the years since Mondo was created, that we have an increasing amount of contributors, and this is numbers from our GitHub repository. So we see there's more and more people using GitHub, contributing to the discussions, contributing to the ontology development, but then also, as you can see on the right, more and more users are more comfortable going on GitHub on our issue tracker and asking for terms directly instead of sending us an email, a personal email. So the community is extremely involved in the development of Mondo. What we also see is that as part of our community, we also have a lot of members who are already expertly curating disease information. So a lot of groups already curate mappings, curate subsets. And so, and on top of that, they're keeping the information up to date. So instead of duplicating effort, instead of competing, we make sure that we leverage this work that is already ongoing, and we collaborate very closely with all of these experts. So we work with experts who create mappings between disease terminologies, experts who curate data, disease informations, and then expert in rare diseases. And we are doing that. We created a process that is simple. I know when we say simple, I should put the air quote here, but it's basically working directly with those sources. Our workflow is such that these external sources create a simple file with the Mondo ID and whatever information they have. And then this spreadsheet is imported into Mondo. We have a lot of QC checks, and everything that passes the checks directly and immediately and automatically added into Mondo. And so anything that does not pass QC checks, we worked with the sources and we report to them what is happening and why we did not include the data. So there's this back and forth loop between the external sources and the Mondo team. As I mentioned, the system is very simple. So all the external sources need to do is create a spreadsheet and we get it. And so because it's simple, it's extremely scalable. And so we can have as many external resources that we need and we want. In addition, every single piece of information in Mondo has provenance of the information. So when all that information that comes from external resources do have a source to report where this annotation came from. So the sources are credited for all the work that they have done. As of for now, we have 6 major sources who maintains information on the site. We get a lot of database cross-references. We also get a lot of subset annotations. We get some links to those other curated disease resources. So this back and forth. We also get a lot of synonyms because we all know that everybody has a favorite name for every single disease. So we also have synonyms. We also work with this group to make sure that we get what they quote unquote preferred name for the disease. We get this, and then as I mentioned, we make sure we have provenance and credit all of that information. One really good example of this process is what we do for rare disease. So we maintain our rare disease subset using this workflow. Our rare disease information came mostly at this point from those 3 major resources, OrphaNet, Nord, and GARD. And we get lists from those sources. We bring them into Mondo, making sure we keep provenance of the information so that we can not only refer to these resources, but also because in the rare disease world, a disease might be rare in the US, but it might not be rare in Asia. So we wanted to make sure that the users have a way to even choose what they want to see when they talk about rare diseases. So we get our rare disease subset from those 3 authoritative sources. Just for completion here, we also do very manual curation if there's a disease that is reported as rare and it's not yet in one of those ontology, one of those sources. We add them to our rare disease subset, and we also leverage ontology inference. So our rare disease subset in Mondo includes all of those resources. And as I said, we keep provenance for this information. So if you look at any of our Mondo terms, you can see that this disease was considered rare in Orphanet, in Nord, in GARD, and so it's in our big picture rare disease subset with the source for the annotations. Other diseases like this one has been reported as rare only in GARD. So again, any users can come to Mondo, get the rare disease, and decide for themselves which rare subset they need to use based on the use cases. But also from the Mondo perspective, we don't have to keep up with all the countries and all the resources to know what is rare at this point in time. So what I've shown you is how we basically rely on our community, and we build this community to make sure that we can keep Mondo up to date, and we can look at the long-term and sustain Mondo. We have this workflow that I showed you that is automated, quality control, and scalable, and very simple to set up. And people who have done it told me it was simple, so it's not only me. Also, it decreases the number of effort duplication, which is really important, especially nowadays. And I have to say, it really increases the sense of community, because now all of those other resources really feel like they have a stake in Mondo and they want Mondo to succeed, and they feel part of the group. So this is really exciting. This is a big thank you to everyone and our whole community. On the left here, you have our current core team. And here, a list of some of our major contributors. Every person, every group here represents a lot of people. So this list is probably and definitely not complete, but yes, we're really, really thankful to our community. So thank you for listening, and I think we have time for maybe one question. Thank you.
Thank you, Sabrina. We do have time for one question. Does anybody?
Yeah, thanks for that talk. That was really nice. So it was great to see the growth in your community of contributors. But one of the things that was interesting on your graph that you showed on the right, which was the number of contributions, you had a really big growth in contributions in 2024. Did you do something differently? Is there something we can learn from what happened in that year? Yeah.
So what I think happened is that we had more outreach. So we started outreach calls so we can talk to people directly, and people really understood how to go to GitHub and create a new GitHub issue. Before that, like those 2 years before the 2024, so 23 and 22, what would happen is that users would email me or would email Nicole and say, I want this new term, and then we would add that that request in the ontology, and then users might come and discuss. But I think 2024, there was a lot of outreach, and then a lot of issues we get from the group ClinGen. They also train the curators and the working group much better, so that they were more aware and felt more comfortable, but getting the issues directly themselves into the GitHub. So it was training and outreach, really.
Thanks. That's great.
Thank you.
Thank you so much.
Thank
I
you.
just have a comment. Not just a comment. No, because of what you were saying, this teaches all, I think, how we are training our community, how we are involved in our community, and just be aware that it's not just our community. We have people that work in many of resources, and this is something that clearly shows the gain in that effort.
Absolutely. Thank you. Yeah, this is a great comment. And we see that, we see, even the awareness of using standard, which I think has become so obvious to all of us. we still have those talks where we really have to explain, this is why it's important. And you change people's behavior and people's minds. So yes, it's a time investment, but I think it's a good investment.
It seems to be
have.
worth it.
Yes.
Okay, thank you, Sabrina.
Thank you.
Tarcisio.
Okay, yeah. Thank you very much for, I mean, it's a really nice opportunity to be here in the conference, and like I'm having a really great time. Then it's really nice to meet you all. My name is Tarcisio. I'm from the Swiss Institute of Bioinformatics. I'm co-leading the knowledge representation unit there. Now, actually, today I would like to talk about our work entitled the Earth Metabolome Initiative Ontology that has been built in the context of the Earth Metabolome Initiative Consortium. As you know, like many in life, actually, you can find chemical compounds everywhere. Chemistry defines species, but not only that, but also the interactions among those species. And it's really important to know that, because with that, it's easier and it's better to understand nature, but also how to heal ourselves, like for example, for discovering new drugs. But one interesting fact is that less than 1% of plant metabolites have been characterized so far. That's really low. Then it means that there's a lot of opportunity, a lot of work to be done yet. And if you don't know what a metabolite is, it's a small molecule that is an intermediary or an end product of metabolism. And in this context, to address this issue, we have started the Metabolome Initiative that we aim at profiling the metabolic content of all currently known species on Earth with three main objectives that are to explore and understand the chemical foundations of the biosphere, but also the benefit of human society and to protect the biodiversity as well. But as I mentioned, like this, we're gonna have, it's already start to be the case, but we're gonna have like a vast amount of chemical diversity data being produced. Then in this context, actually, knowledge representation can play a key role to capture and contextualize and structure this chemical data. And is in these lines that we start to work in this Earth Metabolome Initiative Ontology, mostly for structuring and organize the data that is being produced, but also will upcome in the next years. For doing that, these are some examples of kind of vocabularies that have been used for defining this ontology, for building it. We explored like several general purpose and domain-specific ontologies to design a kind of a framework to describe chemical compounds like natural products and their related data. One important fact is that we also explored like ontologies that go beyond life science and chemistry. Like there are other fields, like for instance, there is some catalogs like the Link in Open Vocabulary catalogs, the link is there. And why we did that? Because we believed also that in other fields, we have similar things that we want to describe, to represent in terms of knowledge. Like for instance, here, we, in a way, repurpose the already well-established W3C sensor observation sample and actuator ontology for describing and representing the sampling and observation data that comes out from the Metabolome Initiative. We also accommodate and harmonize different kind of taxonomies, because it's really hard to have just one ground of truth in terms of taxonomy in the context of the metabolomics. Then, but not only, but also for chemical classification. Then, for instance, we have defined by using like a discuss vocabulary and data schema, the natural product classification that can be used as a taxonomy kind of for natural products. We also reuse some other standards for quantities and units like crudity, but also in terms of taxonomy, we always rely like in this cost kind of based schema for representing it. Like for instance, for the open tree of life, we also use this cost for representing it, and the link that I show here, the Zenodo, it's has this open tree of life representation of SCOS. That's once we have a new release of open tree of life, this is also updated afterwards. Then here, in that link, you can see in the persistence URL, you can see the documentation about this ontology that has been built. Currently, the latest version also includes data modeling for traits and interactions. And we are using, for instance, the relational ontology for classifying the types of interactions among different targets and source organism, essentially. The ontology is also available in the ontology lookup service on ABI. Then based on that, we, in the context of a project that we have been started like 2 years ago, the Swiss Botanical Garden Initiative, that they produce a lot of data. Then we built, it was a nice use case for applying the ontology that we are building. Then we built a knowledge graph based on this, on the AMI ontology. This graph and the methodology and so on that's been used for building it is available in that GitHub repository, and the Sparkle endpoint is also online. And we applied all the tools that our team are developing, like our Sparkle Query Editor that makes possible to do a kind of a smart autocomplete that only, for instance, properties that are stated for given concept in the data will be shown. We also, in a different study that will be appear in the Giga Science journal, there's the preprint there. We also interpret like questions, scientific questions by the different users and the corresponding in Sparkle queries to solve those questions as a kind of a metadata. Then actually those questions are also stored along with the data of the knowledge graph. Then by doing that, for this query editor that you have developed, we can just fetch the questions and show it directly in the interface from the graph. We also have a class overview that, for instance, here we only show the EMI, the Metabolome Initiative Ontology class that's being used and stated in the graph. And this can be also used to other and applied to other knowledge graphs as well, this tool. And finally, we also have like another tool that is for translating questions in natural language into Sparkle queries, the chat.exposé.org, that the user of the question, the answer of the questions of the user is not generated by the large language model, but by the execution of the query over well-structured and curated data that comes from the knowledge graph that has been built in the context of this Metabolome initiative so far. Then to conclude, one point that I would like to stress is that it's really important not only to look at our own garden in terms of knowledge field and so on, but also more broader. Then can be, for instance, we could apply like kind of ontologies and repurpose from other fields in our own fields of interest. And one important thing is also to find like a lesson learned, at least that also comes out from this Metabolome initiative, is to find a balance between abstraction and precision, specificity, in the sense that more abstract we are, it's easier to interoperate with different sources from different domains, but we also lose in precision. Then we have to find a balance. And the tools that I showed, like the Smart Query Editor, the chat.exposé.org, it's everything is freely available in open source. And the ontology is like CC0. And we would like to invite the community, the Biocuration community, if there is any interest to participate in this initiative, please contact us. And finally, I would like to acknowledge my team members, mainly from the knowledge representation unit, Anna Simmer, that's it's my colleague in Zurich that's colleague in the unit with me. Vincent, that's our R&D engineer, software engineer, and also, of course, all the Metabolome initiative members, especially Pierre-Marie and Disha Tandon, and our founders. Without them, I wouldn't be here. Then it's really good to recognize them as well. Then thanks very much. Then if you have any questions, please, then I'll be glad to answer.
Any questions? I see one hand raised. Thank you.
Cool resource. What prefix would you like for this in the bioregistry?
EMI.
EMI. Okay, done.
I expected Charlie to say something about chemistry, but he didn't. So metabolomics across the entire earth is like a lot of
Yeah.
chemicals, right? So, I mean, KEBI, which is the chemicals for biological interest, is already like ginormous.
Mhm.
And your scope is even broader than theirs. So I'm
Yeah.
wondering how you manage like volume in your ontology.
Yeah, okay, to be more precise, in the case for the Earth Metabolome Initiative, the ontology here, we don't seek like to precisely represent like a chemical entity, like it's done by KEBI. Then, for instance, we use actually KEBI in this context. But here, for instance, we will have a lot of information that comes from mass spectrometry data. And for that matter, we try to represent those data that afterwards could be interpreted and link it to more semantical information that comes, for instance, from KEBI. But it's like something, it's like they're complementary, they're not like superposing. And yeah, it's true that the amount of data, it's really enormous that to be generated yet, because so like if I remember well, I read in some article, like I think only, I don't remember exactly, but the number, but it was the chemical compounds found, like the natural products found so far, like they're really, really low. Like even the plants one, like 1% is almost nothing actually. Like.
So this was very interesting. One question I have is about mixtures, because a lot of the natural stuff is actually coming in mixtures form. So how are you handling that? Let's add more to the chemistry questions, because I think chemicals are an unruly bunch.
Yeah, I cannot answer precisely because my background in chemistry is close to zero compared to yours probably. I'm a computer scientist by training. But I believe that in the consortium, like, of course, there is the concern. And probably these, at least from the data that we have so far that I have seen, this is probably done before we inject it like in the knowledge graph, actually structure it. Then this work of trying to handle this mix of chemical compounds and so on, then it's done, I think, before even like the data that we use it for structuring the data here in the knowledge graph, actually.
Right. Are there any questions? I don't think there are. Oh, yeah.
So are you intending to be part of the Metabolome Exchange project and use all the data standards for it that they're developing? Well, the Metabolome Standards Initiative are developing.
Sorry, I didn't get the question. Actually.
Are you intending to join the Metabolome Exchange Consortium and use the standards that they're developing, the MSI, the standards initiative?
That it's I mean, it's like there is like the consortium that you mentioned that I think it would be really important to be aligned with. This is for sure. This is something that I can bring also to the community, because this is true that is likely more like European centric now. We are actually. This is the problem. And then but yeah, the idea is to expand it. Then then it's not
give you
like
access to a lot more data, because
of course,
you know, access to all what's in metabolites and all the other
we currently have like people that are involved in this consortium. They have like more or less like 4 different projects across Europe. But again, they are really more like European centric. Then I think this is also a problem that we should address in the next. Thanks.